Good evening, everyone. Thank you so much for coming out to tonight's talk. Before we begin, I'd like to start by recognizing the traditional, ancestral, unceded territory of the Musqueam people here at Point Grey campus, and we thank them very much for their hospitality. Tonight, I have the great pleasure of introducing this year's Cecil H. and Ida Green's visiting professor at UBC, Dr. Imogen Coe. It's always great to host a scientist at Green College. We don't get as many as would be nice, so it's a particular pleasure to have Dr. Coe share her insight on a couple of different topics here at Green College this week. If you were here on Monday, you will have heard her talk on equity, diversity, and inclusion in Canadian academia, and tonight we are here to learn about some of the science that she does. So I'll just give a little bit of a brief introduction on her background before we get into the really interesting part of tonight's lecture, the science. So Dr. Coe studied for her BSc honours at the University of Exeter in the UK before coming to the University of Victoria, out from rain to more rain on the west coast here, where she investigated the molecular biology and biochemistry of the biochemistry controlling the endocrine system of several fish species. However, it wasn't until her postdoc at the University of San Francisco and the University of Alberta that she really hit upon an area of research that she would eventually build her lab around. At UCF, she worked on understanding how ethanol affects neural cells, particularly how ethanol affects the transport of adenosine across the cell membrane and how this changes upon chronic exposure to ethanol. Perhaps you can ask her what is happening to your brain cells as you enjoy a drink at tonight's reception. At the University of Alberta, she started studying the membrane proteins that are responsible for transporting nucleosides like adenosine across the cell membrane, and really getting into uncover the genes that are responsible, working on cloning and identifying those genes that code for the proteins that transport the nucleosides across the membrane and understanding their biochemistry. Following her postdoc, she was recruited to the University of York, or York University I suppose it's called, where she started her lab, which continues to explore the biochemistry of these fiendishly difficult membrane proteins. And her research not only has implications for understanding basic cell biology of how cells function, but also lends insight into our understanding of how many anti-cancer, antiviral, and anti-parasitic agents work. Evidently, running a lab and teaching was not keeping her busy enough, because in 2012, she was recruited by Ryerson University as their founding science dean. In addition to her work as a scientist, educator, and administrator, she has worked tirelessly as an advocate of equity, diversity, and inclusion in academia and beyond. Tonight we're here to learn about her science, and she's already spoken on this earlier in the week, so I won't say too much about that. But I will just say that she has done a lot of work in policy and communication with her TED Talk, numerous radio interviews, and in 2016 she was announced as the Women's Executive Network of Canada's top 100 most powerful women in Canada. Please welcome Dr. Coe to the podium.
Sorry. Let's see. I might just walk around with this. Oh, okay. Hopefully everybody can hear me. Is that okay? Yeah. Oh, is it? Shall I take it off? Or just... Okay. Okay. Sure. So this is okay. Okay. Use my inside voice. Okay. So thank you for that very kind introduction. And I do send you my thoughts and prayers for dealing with fiendishly difficult, we tend to describe them as evil membrane proteins, actually. I think you're being quite generous there, because they can be very difficult for people to work on. So it's a great honor to be here for the Cecil H. and Ida Green professorship. It's always a real privilege when you're invited by graduate students, and so that's very, very special, and I'm very, very grateful for that. I did give a talk on Monday, and I'm going to talk a little bit about my work as a scientist. So, and this is where I do my work at Ryerson University in downtown Toronto, or where my lab is actually located, which is at St. Michael's Hospital, which is right next door to Ryerson in the research wing there. Okay, let's try that. We always acknowledge also where we do our work, and Ryerson is in the heart of Toronto. Toronto is in the dish with one spoon territory. That is a treaty that brought together the Anishinaabe, the Mississaugas, and the Haudenosaunee to share the territory and protect the land. Subsequent indigenous nations, peoples, Europeans, and newcomers have come into this treaty. I don't believe that they were always invited, so this is a living document. We update it as we become more self-aware around our relationship as settlers and colonial peoples with First Nations. But the principles of the treaty are principles that we should all embrace, which are the spirit of peace, friendship, and respect. So I acknowledge the people that have lived in the Toronto area for tens of thousands of years, and we are always looking for ways to be much more conscious and mindful of their ways of knowing and learning from their work that's gone before. So I'm going to talk today about the work we do on drug transport proteins. And when I first started working in this field, I would go to conferences like the American Association for Cancer Research, one of the biggest cancer research meetings in the world, and there would be drug companies and research scientists, and they'd be developing new drugs, new anti-cancer agents, and they would show diagrams like this, that there would be drugs, and there would be an arrow, there would be like a cell, a round thing that was a cell, and there would be an arrow, and there would be a statement that said drugs get into the cells, and then they would show some data, and they would say the drugs do wondrous things, and there was never really any recognition that doesn't reflect on what happens in reality at all. And the reality is that, as we all are, I hope, aware, the cell is an incredibly complex, incredibly dynamic, incredibly rich and amazing structure with all sorts of elements and components that exist both within the cell, but also importantly at the cell membrane. There are complex subcellular compartments. There are things moving in and out of the membrane. The membrane itself is actually very complex, and it forms a barrier to access for things getting in and things getting out, whether those be things that your cells need, like glucose and amino acids, or they could be waste products that your cells want to get rid of. The clinical relevance of what happens at the cell membrane is really significant, because membrane proteins, so things that are sitting in the membrane, represent a significant proportion of drug targets. So drugs that we use clinically are often targeted to receptors or to other proteins that are sitting in the membrane, and that's a significant component of the drugs that we are using in the clinic. So we need to understand how that membrane protein community actually works, what regulates it, how those drug targets work. And proteins that sit in the membrane also are very important as routes of entry for drugs. So not only are there proteins that are targets, there are also proteins that are gateways or doorways, or somehow facilitate the entry of drugs that are very important in the clinic. So we need to understand how things get into cells. We need to understand how drugs get into cells if we're going to understand clinically relevant kinds of problems or develop new drugs. Now, I put this slide up, and it's deliberately meant to be impossible to read, because I think it's really important to recognize that there are huge numbers and a huge diversity of these transport proteins that exist in all of your cells. And up at the top here are 3 kinds of cellular settings your intestines, your liver, your kidney, which have in their membranes around either the side that's facing, for instance, in your intestine here, the gut, and on the other side of the cell, the side of the cell that's facing the blood supply, different sets, different combinations and permutations of transport proteins that are all doing different things, usually in coordinated fashion, and they could be moving things in one direction or in both directions, but they're certainly acting to ensure that those cells get what they need, get rid of what they need, and we also use them as routes of entry for drugs that are important in the clinic. And you might be able to just squeeze out some drug names that you recognize there. Metformin, that's used in the treatment of diabetes. Statins, there are some other methotrexate, some other drugs that people might have heard of. And these all rely on these transporters or these carrier proteins or these gateways in order to get in. If we don't have those gateways or those transporters in our cells, it's very difficult for those drugs to be effective. All of these transport proteins have evolved to transport some endogenous compound, some naturally occurring compound that our cells need, but we now use them as gateways for getting drugs that are used in a clinic into cells. So the drugs, so the transporters that we work on in my lab are called nucleoside transporters, and they are families of similar transporters, and you can identify them out of the human genome. They've been cloned, we can study them, and we know roughly what they look like. So for my nucleoside transporters, there are two non-homologous, two families that both do similar things, but they have they're not similar, they do similar things, but they have no structural or sequence similarity to each other, which is interesting in its own right. And we predominantly look at this family, which is called the solute carrier 29 family. There are 4 versions in humans. You all have ENT1, 2, and 3, and 4. They're called equilibrative nucleoside transporters. That means that they take a substrate and they simply facilitate its movement down its concentration gradient across a plasma membrane or across a membrane. And they will transport nucleobases and nucleosides and nucleoside analog drugs. Nucleosides and nucleobases are important building blocks of nucleic acid. And when a cell is dividing, it needs to make lots of nucleic acid. It needs to make another copy of the genome. It needs to make more DNA because it's dividing, and every generation of cells needs to have a copy of the genome. So these nucleoside analog drugs are effective because they interfere with the synthesis of DNA. They interfere with the ability of a cell to make more DNA. And the places where you might want that to happen, you might want to stop that happening, are places where you get or situations where you got uncontrolled cell division. So those would be situations like cancer, where cells are dividing in an uncontrolled way. They're making a lot of DNA. So if you can interfere with that, then that's a way of treating uncontrolled proliferation. Viral infections also are situations where you get a lot of DNA being made. Viruses continually are replicating and dividing and multiplying. And parasitic infections do the same thing. They tend to hijack the cellular machinery and make the cells divide. So these are all situations where you want to stop DNA synthesis or RNA synthesis. And one way to do that is to give people drugs that are similar but limiting in terms of their ability to actually, for cells to use them to make more DNA. So I was part of a team that cloned the very first one of these family members, ENT1, back a long time ago at the University of Alberta. And for many decades, we really didn't know much more than this was the structure of our transporter, a unique 11 transmembrane domains. So the protein actually weaves its way back and forth across the membrane 11 times. We didn't know anything more about that kind of structure until just very recently this year in the summer, when a 3-dimensional crystal structure was actually finally reported. And that is quite a breakthrough because these proteins are very, very difficult to work with biochemically. So they're very difficult to study. They're very difficult to purify. They're very difficult to manage and measure biochemically. So this was a considerable breakthrough. We can now look at how this transporter works. There are 4 versions of it. They all have the same more or less structure, different sequences, and they transport nucleosides and nuclear bases. I just want to mention that there is another family that is also important in drug transport, but we're not studying that one, and that family has slightly different characteristics. So back to Biochemistry 101, what does a nucleoside look like? Just to remind you, or to inform you, a nucleoside is a ribose sugar, so there's a sugar structure and then a nitrogenous base structure. And there are kind of 2 flavors of nucleosides. There are the purine nucleosides and the pyrimidine nucleosides. They're differentiated on the basis of the structure of that nitrogenous base, 2-ring structure or a single-ring structure. And these then form the basis of building blocks for DNA. If we modify those structures a little bit, if we chop off that ribose sugar and just focus on this component here, that is a nitrogenous base, nuclear base, and my transporters will also move those things across membranes. That can be quite important because there are drugs that are based on that structure. And if we add a phosphate group on to the ribose sugar here, then we get a nucleotide. And we can add 1, 2, or 3 phosphates, and we'll get a nucleotide monophosphate, a nucleotide diphosphate, or triphosphate, and it's the nucleotides then that are strung together to make DNA. So a strand of DNA is a whole collection of nucleotides strung together. And these structures have been known for a long time, and indeed purine and pyrimidine metabolism has been well described and is fairly well understood. The pathways are complex. All your cells are involved in all of these pathways all the time. It's going on in all your cells. You don't feel it, but your cells are really, really busy, and you can see that there's a very complex set of pathways there involved in continually turning over and using different components in these pathways. There are a number of clinical conditions that are the result of dysfunction or imbalance in some of these pathways. They're the things that are actually in little rectangles here. And one that has been known for many hundreds of years is an imbalance in purinergic metabolism that leads to gout. So if you have an imbalance, yes, does it feel like that? Yeah, if you have an imbalance in purinergic metabolism in a certain pathway, you can get a buildup of uric acid crystals. They'll precipitate, and they tend to precipitate probably just because of gravity in the joint of the big toe. And this is a picture cartoon from a couple of hundred years ago, and you can see there's a little monster like chewing on that toe joint, and if you know people that have had gout, or if you had gout, it's incredibly painful. And that's a consequence of an imbalance in purinergic metabolism. So one way to treat that is with a drug called allopurinol. Allopurinol is a nucleobase analog of a naturally occurring nucleobase in your system called hyperxanthine. Hyperxanthine inhibits uric acid production, and so allopurinol is used to treat gout. And it's an old drug. It's been around for a very long time, and it's an effective drug. And so there are drugs that have been known for a long time. There's a naturally occurring nucleoside that we all use a lot, which is adenosine. And I've got it kind of described here as the oldest drug because adenosine is evolutionarily very ancient in terms of being a signaling molecule, and it actually signals to cells, particularly in your brain and your cardiovasculature, to say maybe you're getting a bit low on oxygen, or your blood supply is a bit compromised. And adenosine actually triggers responses in cells to resist kind of cellular insult. And adenosine is actually given clinically in situations in cardiology or nephrology as a way to actually boost cell survival and try and help cell systems, cardiovasculature, to resist drop in oxygen or heart attack, that kind of thing. So adenosine is naturally occurring, and it's actually a very old drug. It's almost as if your body uses the drug itself. And the adenosine, this is just represents the adenosine transporter, or the nucleoside transporters that I work on, which do clear adenosine from the outside of the cell. They bring it into the inside of the cell. And if you have a drug that blocks the transporter, that stops this process happening, you actually get an activation of adenosine, or possibly the compounds, ATP or ADP, that get broken down to adenosine, will act through other membrane proteins, such as receptors, to stimulate pathways that are very good for, or very helpful in terms of resisting cellular stress. So there are also drugs that are involved that are used clinically, dipyridamole and dilazep, that are used clinically they're targeted to the transporter, that block uptake and are important in promoting sort of purinergic signaling. So all of these proteins that sit in the membrane then are part of another complex that we call the membrane purinome or the membrane proteome, and they're involved in cellular physiology. And my lab over the years has done a lot of work on understanding how the transporters, how those adenosine, those purinergic transporters, modulate the effect of endogenous, naturally occurring compound nucleosides in various locations. Particularly, we've done a lot of work in the cardiovascular system, but all through your body, these equilibrative nucleoside transporters are working all the time to make sure your cells are working optimally. Now, ultimately, fundamentally, I'm a cell biologist, and we are interested in really how, this is another representation of these little transporters, how these transporters are regulated within the cell. So we've looked at a number of different aspects of the transporter, from protein synthesis to different kinds of post-translational modification. How does it get sorted to the cell membrane? There are other pathways such as phosphorylation. The phosphates are added or taken away that seem to regulate the action of the transporter. And then there are also other proteins that come in and interact with the transporters and change the way they behave. So a lot of cell biology, just trying to understand how these things work, where they're found, what they look like, what other things regulate them, because we just want to understand the basic physiology. But what I wanted to talk about today was some of the more recent work that we're doing about how we can use some of that knowledge to improve the way we use some drugs in anti-cancer therapy that get in through these transporters. So this is just to remind people again that here's our transporter. We give people drugs, anti-cancer, antiviral, anti-parasitic drugs, and they get into cells through our transporters. And then there's a series of metabolic enzymes that turn them into nucleotides, and then they get incorporated into DNA. And because they've been modified, they interfere with that particular process. So many anti-cancer drugs, and this is just a very few, just a couple that are represented here, have been designed on the basis of the basis that they look similar to compounds that cells are used to seeing, and they look similar to compounds similar to nucleosides that cells use to synthesize DNA. So here we have something that is naturally occurring. You will have deoxycytidine in your system, and this is an anti-cancer drug called Ara-C that you can see looks very similar to deoxycytidine. There's a slight modification over here. There's another anti-cancer drug that I'm going to talk about more later called gemcitabine, which is also an analog of deoxycytidine. But you can see, I hope, that there are two fluorine molecules here on this carbon, on the ribose ring, that are highly charged molecules. So while the cell sees this as looks a lot like deoxycytidine, when that nucleoside gets phosphorylated to a nucleotide and the cell tries to incorporate it into DNA to make more DNA, those two highly charged fluorines interfere, and the cell actually can't use it the way it thought it could, and that cell dies. Fludarabine is another drug that is used in the clinic. It's a purine analog. And then there are nucleobase analogs that are also used clinically. 6-mercaptopurine, used a lot in certain conditions, and 5-FU. And there are literally thousands of nucleoside analogs because we continue, companies like Eli Lilly or Glaxo Wellcome are continually synthesizing new variants on nucleosides in an attempt to continue to generate new drugs. But whatever that drug looks like, it still is dependent on the transporter to get in. So it still needs to get into a cell. It can't just like drift across the membrane. It needs a facilitated transporter to get in. And these drugs have been around for a long time. And I just want to sort of make reference to a hidden figure here, Trudy Elion, who shared the Nobel Prize with James Black and George Hitchings for innovative rational drug design that started back in the 50s. That instead of just doing trial and error in terms of drug design, they actually looked at purinergic metabolism. They looked at pyrimidine metabolism and looked at the endogenous compounds and then tried to design drugs based on what we understood about naturally occurring compounds. She's one of the very few Nobel Prize winners who didn't have a PhD, and the reason that she didn't have a PhD is that she couldn't get graduate student funding in support of graduate studies because she was a woman. So I just thought there was an interesting, particularly because the graduate students invited me here, that you know, fortunately she found sympathetic people that she could work with, very, very talented chemist, but you know, she was you know, sort of one, I think she got turned down 15 times for graduate funding until she found somebody who would support her. She worked as a high school teacher and stuff. Tremendous talent that might have been lost had it not been for people that were supportive of her. The number of drugs that were developed as a consequence of the work that Trudy Elion and other people did is truly remarkable. 6-mercaptopurine, Allopurinol, which I told you is used to treat gout, AZT, used in the treatment of HIV/AIDS, acyclovir is an antiviral, another antiviral, pyrimethamine used in the treatment of malaria, and a whole host of other drugs used in things like to prevent organ transplant rejection and that kind of thing. So a consequence of the work that people like she and others have done, huge numbers of lives have been saved and lives have been improved. So we're interested in the nucleoside analogs that she and others received the Nobel Prize for, and we're interested in how those transport proteins might influence the efficacy of those drugs. And my lab was actually one of the first to show that different people have different profiles of those transport proteins. And that's kind of important because the efficacy of the drugs is related to the drug getting in. So if you have more of the transporter, maybe you can get more of the drug in. So these are different patients. Each one of these bars is actually an individual patient. So patient 1 has very high levels of ENT1, much lower levels of 2, about the same level of 3, and very little of these other transporters. This patient 5 has very high levels of ENT1 and ENT3, nothing of these others. So these profiles can start to give us some ideas about maybe some people are better candidates for nucleoside analog drug therapy than other people are. This patient, number 4, has sort of lower levels. Maybe they're not such a great candidate. And this is very important to understand how people might relate, particularly to the anti-cancer nucleoside analogs, because the side effects can be so incredibly devastating. And in fact, we know that ENT1, that first isoform, its presence in cells is both sufficient and necessary for a response to a particular drug called gemcitabine. So we know that if you have high expression levels, if you've got a lot of this protein in your cells, we know that you need to have that, and that is related to how well you will respond to an anti-cancer drug called gemcitabine in a couple of different situations, lymphoma and biliary tract cancer. So we actually are focused on this particular drug. We could work on lots of drugs, but we're focused on gemcitabine, a nucleoside analog drug, which is an analog of deoxycytidine. It's been used in the clinic for quite a long time, so it's considered quite well established. It's an old drug. It's relatively well tolerated. And it's still the first line of treatment in pancreatic cancer. It's often used in combination with other drugs, but it's still predominantly, typically, the first line of treatment as an anti-cancer or as a chemotherapy agent. Now, pancreatic cancer is a particularly challenging disease. These are the 2017 data from the Canadian Cancer Society. You can see approximately 5,500 Canadians diagnosed. In 2017, 4,800 Canadians died of pancreatic cancer. It's a challenging disease. It's often discovered late. It's difficult to treat because the pancreas sits behind the stomach. It's not an easy organ to get to for surgery or for radiation. So it's a challenging disease to treat, and we really need much better treatments. So we are looking at how can we improve the use of gemcitabine. Are there sort of novel, innovative ways that we can improve the way that drug works? And one of the reasons to do that is that drug is well established. We don't have to go through with a new drug clinical trials, figuring out whether it's going to work or not, and spending a lot of time working that out. It's a well-established drug. It's a good drug in that it's relatively well tolerated. The side effects are not as bad as maybe some other drugs, but the side effects are still significant. So the things that we're interested in, the questions that we're trying to answer are: can we develop more targeted delivery? So this drug is typically given systemically, so it goes into the blood system through IV. So it's going all around the body, but you're actually trying to treat one set of disease in one location. Can we target that better? Eventually, pretty much all chemotherapeutic agents, whether they be anti-cancer or antiviral drugs, cells will develop resistance. So cells will develop other proteins that will pump drugs out or will metabolize drugs. That is what chemoresistance is. Cells become resistant. The drugs stop working. And can we figure out new ways to use drugs that are already on the market rather than trying to find a new drug? And because I'm located at Ryerson in downtown Toronto, I've had the opportunity to do some what is very interesting and cool for scientists, work with scientists and engineers in other disciplines and come together with new ways of thinking about maybe solving a problem that's been a problem for a long time. So working with medical physicists and biomedical engineers at St. Michael's Hospital in a Institute for Biomedical Engineering and Science and Technology, which is a combination of St. Mike's and Ryerson coming together. I'm basically providing a context where clinicians and biomedical engineers and medical physicists and cell biologists can come together and look at very applied, very translational kinds of research projects. And so I'm working with Dr. Mike Kolios here, Dr. Scott Tsai, and Dr. Rafi Karshafian, using new approaches to try and figure out, can we improve the efficacy of gemcitabine? And all of the people here are using now new approaches to try and mimic the situation that you might find in a tissue or in a cell by doing what's called lab on a chip. So basically, we grow cells in a system where we can flow media over them, so it's more like cells might experience in a pancreas or in a blood supply, rather than the traditional way to study sort of cell biology, which is by growing the cells in dishes and then looking at them. So using a lab on a chip approach, and we're using ultrasound and microbubbles, which is probably not something that many people have heard of before. Maybe you have. So this is a technology that's been around for probably a decade. And it's a very interesting technology that the biophysicists have really developed that involves giving people by IV solutions that have tiny gas-filled bubbles. They're called microbubbles. They have like lipid, like fatty surrounds a gas, like a gas bubble, and they're very well tolerated, they're very inert, they get excreted out through the kidney. But the advantage of using them is that if you expose them to ultrasound, you get what's called, well, you get expansion and contraction, the ultrasound, just ultrasound like you would use for looking at a baby in a pregnant woman or looking at a joint. That kind of ultrasound in a targeted fashion causes an oscillation, causes the gas to expand and contract, and eventually you get something called cavitation. The bubbles will burst. And if you have these bubbles within the vasculature, within the blood supply, particularly, for instance, if it's going through a tumor, blood supply is feeding a tumor, and you target the ultrasound to that location, so over the pancreas, for instance, then you will get, depending on how you modulate the ultrasound, you can get bubbles expanding and contracting, and they can disrupt the cells around where that's happening. They can disrupt the tumor. If they burst, they can actually kill cells. So they can actually, this is actually a cytotoxic treatment, and it's non-invasive, and it's doesn't have side effects because it's very targeted. So one of the things that a number of people are doing, including people at Ryerson, is looking to see if giving people drug, so you put drug into the system, and then you give them microbubbles, and then you apply ultrasound. Can you kill the cells and also get more drug into the tumor and have a combination kind of impact of drug plus ultrasound microbubbles? And it actually turns out that sometimes you get synergistic effect. You actually get more cytotoxicity as a consequence of this combination approach. So the mechanism of the approach is that you can get more drug into a tumor because you break through vascular cells that sometimes are limiting drug uptake. You also cause tiny little pores, tiny little holes in the cells that allow drugs to get taken up. And the combination of these things leads to then a combined cytotoxic effect. Now, nobody really had ever asked the question, can you use ultrasound microbubbles and nucleoside analog drugs? So people had done this ultrasound microbubbles with other drugs that are used in cancer treatment and other kinds of clinical settings, but nobody had really ever looked to see if this very big class of anti-cancer drugs, or just one of these anti-cancer drugs, nucleoside analog drugs, in combination with this biophysical therapy, would actually have a synergistic or an additive, or maybe they would compromise each other effect. And we asked this question, and we were, and there was a rationale to ask this question, because a very small clinical trial had basically asked this question as to whether this combination of ultrasound microbubbles and gemcitabine would extend life in terminal patients with pancreatic cancer. And so this is a figure from that study. Here is a patient, obviously. Here is the ultrasound wand that is focused over the pancreas. The patient has been given drug. Here, time 0, they're given drug for a number of minutes, 20 minutes up to 30 minutes, to allow the drug to go through the system. It's given into your veins. It goes through your system, and then ultrasound and then microbubbles, and then they are exposed to ultrasound only in the location where that wand is sitting. So there is targeted ultrasound going to the pancreas. There is drug and microbubbles flowing through there. The bubbles will be oscillating, they will cavitate, they will break, and that will drive drug into cells, and the gemcitabine is also killing cells at the same time. So the result from this very, very tiny clinical trial, it was only about 9 patients, and they were all enrolled because they were terminal, suggested that there was a positive impact, that it did seem to lead to a slight reduction in tumor load. There was an extension in lifespan, and so it suggested that there was a rationale for actually looking at this. But nobody had really looked at the underlying cell biology or biochemistry of it, so we decided that we would do that. So we did this in the lab. We took cells that are pancreatic cancer cells. We put them into a system where we could expose them to ultrasound. We mixed those cells with microbubbles and we tested to see if we could regulate ultrasound pressure, which we could. We could actually increase pressure and we could measure the impact of that pressure. We did a detailed pharmacological analysis so that we knew that we could determine exactly what the IC50 of that drug was. We knew how much drug in those cells was needed to kill 50% of them, so we could measure the pharmacology, so we knew exactly what the combination would be and what we would expect to see if our hypothesis was right. And when we combined those together, we could see that if you look at ultrasound alone, as you increase ultrasound, you kill more and more cells. So this is cell viability. In the absence of ultrasound and no drug, then you've got 100% of your cells are alive. As you increase ultrasound, it's quite effective. You kill cells. Then they're sitting in a little dish, so it's a very artificial system. But if you add drug, you also kill cells. And we started at a concentration where we were using a concentration that was an IC50, so we should have seen a 50% cell death. We did. And when you combine those two modalities together, you actually get an increased cell death. So this line then is gemcitabine at an IC50 plus cytotoxicity. So you actually get an additive effect. Now this is a little different from other drugs where they've seen a synergistic effect, but we got an additive effect. So those two conditions together are still better than each condition alone. And importantly, those two conditions together didn't interfere with each other. So we weren't seeing a reduction in the level of cytotoxicity. So this suggested that, yes, we could manipulate the modalities, we could measure the modalities, and we could actually really investigate this as a potential therapeutic, innovative therapeutic combination modality that might be applicable in a clinical setting. People had already done a little tiny clinical trial. Now the cell line that we used was actually a cell line that was considered to have only moderate resistance to gemcitabine. It was a cell line that you could actually kill quite easily with gemcitabine. And so that might not have been such a great model for testing whether adding another modality was going to be useful, because we can actually kill these cells pretty well with drug. So we then moved on to another cell line, also a pancreatic cancer cell line, which has what's considered to be high resistance. It's hard to kill. It's harder to kill these cells with drug. So that might be a better candidate for a combination modality, because if you're going to give people drug and they have this kind of more resistant type of cells that are causing the cancer, then you want to come in with a secondary modality to try and deal with those more resistant cells. So the questions then we're asking here, and the reason for this is, if you have a cell, if your cancer is more resistant, then you're going to have to use more, you might have to use more drug. But if you have a second modality that you can treat people with, then maybe you can use less drug, and then you'll get, you can use less drug, but you'll get equal impact in terms of the cytotoxicity. Or you could use less or the same amount of drug, and you could target it to maximize impact, and all the time aiming to reduce side effects. So less drug for a more resistant cell type minimizes side effects, or maybe you could optimize the treatment sequence for maximum impact. So it doesn't matter which order you actually give these treatments in, and you can then target your treatment via ultrasound microbubble. And what we found very briefly is that with these more resistant cells, yes, if you just grow them in the lab and you just let them grow and you measure how many of them die over a couple of days, it's only about 5%. So they're a tough cell type. If you treat them with ultrasound microbubbles under the same conditions we'd used previously, you only get about a 20% cell death. Still kills cells, so that's good. If you treat them, we were using a concentration of gemcitabine that we knew should kill 50% of them, and it did indeed, but a high concentration and not necessarily one that you want to use in the clinic. If you use those two conditions together, you saw approximately an additive effect, about 50 plus 19. So you did see an additive effect. If you used the ultrasound and then gave the drug, you saw a bit more. But really interestingly, if you used drug and then came in 48 hours later with the ultrasound microbubbles, you were killing a lot of the cells. So in the absence of those two conditions, you get 5% cell death. If you put them together, you give drug for 48 hours, and then you come in with ultrasound microbubbles after that, you kill almost 90% of those cells. So sequence matters. And that's a really, really important and really promising kind of result that obviously we're following up on. So, and it confirms in the lab what this very small clinical trial that could have been kind of almost random chance, and the significance of the increased longevity or the significance of the reduced tumor load was very small, but it does give us a solid basis now within a sort of a control setting to say, yeah, looks like if you give drug and then you come in with the secondary modality, that's actually a promising approach. The other thing that we discovered, which I think is also really, really critically important and speaks to the resistance that we see in many, many anti-cancer treatments, is that when you treat cells with drug, now these are our Panc1 cells, and they've just been stained with some regular cellular markers, and this is what they look like. Here's most of the cell, and then these are the nuclei stained blue, and this is together. They're kind of cuboidal, they're kind of square, they're these little blobs. But when you give them drug for 48 hours, they start to change. Some of them start to change. And I hope you can see that some of these cells have different kinds of shapes. They're beginning to change their shapes. They're beginning to spread. They don't look like these little squares over here. And what this looks like is something called epithelial to mesenchymal transformation. And these cells are showing signs of becoming resistant to drug. They're also showing signs of transforming into a cell type that is more metastatic. They're showing signs of transforming into a cell type that's more mobile, more migratory, more invasive. We've got other data to show that all of those things seem to be happening. And what that means is that when you give a gemcitabine for a length of time, you kill all the cells that are susceptible, but you actually leave behind a very dangerous, tiny little subset of cells that then can potentially go on and become really problematic. So it speaks then to the advantage of coming in with a secondary modality or a secondary treatment, such as ultrasound microbubbles, because there's no distinction. That's a biophysical approach, biophysical intervention. There are no cells, there's no resistance to that kind of intervention. So our cells after 48 hours, many of them are dead, but a few of them are showing these kinds of signs, and then our potential is to come in with our secondary modality, and then ultrasound microbubble will kill these cells. So that's our new tricks and our old drugs, and we're working on that and we're moving to a lab on a chip approach with the biomedical engineers. The people that really do all the work, as is almost always the case for science, are the graduate students and the postdocs. So I'm very proud to have a very international, very diverse group of students over the years. This is from a couple of years ago, but much of the work now is being done by Bianca, who is doing the cell biology. Sıla here from Turkey is doing the gemcitabine EMT transformation work. Julia did the original ultrasound microbubble work in the lab. This is Latina. She's my longtime research associate who runs the lab for me, and without whom I would not be able to do anything. And these are the people that have paid the bills over the years. NSERC, CIHR, and Ryerson have provided funding in support of research. And our collaborators at Ryerson, a cell biology collaborator in Barcelona, and more recently structural biologists in Toronto that we're also working with. So thank you very much for your attention.